science papers are like buses. You can wait for a decade and none will come along, and then you get 3. So it's fantastic to be here, because this completes the full circle from me for being a postdoc in John's lab, and it's just thanking John and Tom for not only enabling my scientific career, but also for being here today. So I'll be talking about emerging fungal pathogens. I'll be talking a lot about frogs. This is my colleague Claude Mio. He's swabbing for chytrid, the largest frog in the world, the Cameroonian Goliath frog. It's enormous, isn't it? But I'll start further back than that. So if you're sitting comfortably, then I will begin. So it all goes back to this. I was a little 10-year-old, and I grew up in Africa, and I didn't put on shoes for the first 10 years of my life. But this is an ecosystem which has fantastic fungal diversity. At the time, this Timitomyces giganticus was bigger than the one that was in the Guinness Book of Records. So I really appreciated the deliciousness of kingdom fungi. But I wanted to be a parasitologist, because that is what my family business was. So this is a paper by my grandfather, A.C. Fisher, second generation Zambian, a study of the schistosomiasis of the Stanleyville district of the Belgium Congo, published in '34. And in this he described a new species of Bilharzia. So, you know, neglected tropical diseases, went all the way back then. I wanted to be a parasitologist. So I took up this kind of gastrointestinal theme. I worked on gastrointestinal nematodes of rats when I was a PhD student in Edinburgh. And this literally was a shit PhD. Partly because that's how I had to culture my organism. I also learned that it's just, you know, the randomness of science is incredible. This is the number of rats I caught. The black ones are the ones with my study organism in. You can see I was an Edinburgh student, so that was years 1 and 2. 5 and 0. So I was really unhappy at that point, but I went down south and got things to work. And then there was this amazing technique called PCR. Tom could probably tell you a little bit about that. But anyway, you could, instead of using allozymes, which everyone else was using, you could cut these with little cutters and score your alleles that way. So, you know, I learned some pretty nifty tricks about pathogens and genetic epidemiology back then. But, you know, my career really didn't feel like it was going anywhere. And this is a picture of me sitting under a Scottish rock. And you can just see I'm not that very I mean, this is the nadir of the PhD progress. So when I walked past the departmental notice board and looked right rather than left, and saw the job advert for this place in California with this guy, didn't know who he was, I applied. And I got the job, and it took me from out from under the rock to this place here, which was John and Tom's lab between 1997 and 2000, I think it was 2001. And this was just such a fantastic, buzzy environment. There were packs of people passing through. I think I've stolen some of the Bruns lab. But they, you know, all of these people were incredibly influential in my thinking. And they took this really green parasite biologist, here newly arrived, and turned them into this wonderfully flamboyant Castro creature, because I lived in Castro with my young family. My friend Evan's in this room, and you can actually see his there. So, you know, I embraced the local flavour of this place. And, you know, I was presented with a number of options upon arrival, but I plumped for working on Coccidioides immitis. This, you know, it was a pretty scary fungus. And at that point, Vaso Kufupanu had managed to divide it into 2 clades, the California and non-Californian clade. So I needed better markers than Vaso had, obviously, because, you know, she's got a whacking great branch there, but nothing more. So I decided to work with these things called microsatellites. And anyone who's cloned the microsatellites, actually none of you would have, because you would just sequence the genome. But you actually had to go through a business, and it was really quite difficult. And I happened upon this protocol on the internet, the new internet, called How to Get Your Markers Before the First Few Months of Your Graduate Degree Programme is Passed, by this slightly chubby Canadian. So, you know, I thought I'd give this protocol a go, and by golly, it worked. This is the first synthesis of our ACAC probe. I screened a genetic library of Coccidioides immitis, and then the secondary pick, and you can start to see that I've nailed it. You know? And this is just pure pleasure when you see a southern like that. So at that point, we had highly polymorphic markers, and we could drill down into Coccidioides immitis, and I learned a couple of things in Taylor Lab. Firstly, that morphological fungal species are home to deep evolutionary structure, and this is the first species description of a fungus using dust nucleotide characters. Coccidioides persallus species novo, described off the back of SNPs and microsatellite alleles. Now, I also learned that fungal populations are dynamic entities, and that epidemiological processes can be described through the lens of genetic diversity. And through applying these molecular markers to the cocci populations across the New World, we not only saw the species, but we saw all these subpopulations, and then what looked like a recent invasion of South America from the Texas subpopulation. So I really started to feel that this kingdom not only had a lot of kind of interest from the point of view of an endemic pathogen, but there could be deeper processes at play. So my mind was primed for hearing a conversation between John Taylor and David Wake on this fungus that was killing frogs. And that's, you know, that chance favours a prepared mind. That idea was there. So we, you know, at that time, a lot of the kind of stories about fungal diseases impacting more now than they had in the past were flying around, and so we decided to be a little bit more systematic about this. So I worked at that time, and I still do, with Lady Sarah Gersh. She's not a lady, but she should be. She's you know, a very refined creature and a very systematic biologist. And we started to do an analysis of case reports of disease alerts that are attributable to fungi across time. And we found that for both plants and animals, there was this global distribution of alerts. And then we could start to see signals of an increasing signals of more disease alerts in the now than there were in the past. So these as a proportion of all the pro-med disease alerts that there are. So the other classes of pathogens are losing out to fungi, both for plant-infected fungi and animals. Not only this, but they're increasing, where you actually saw a host species that was being decreasing owing to a pathogen, it was more likely than not caused by a fungus, and fungi certainly were overrepresented as drivers of host declines. So they really seem to be having an impact on biodiversity. And so this led to the publishing in Nature 12 of our Fear of Fungus paper. There are reasons to be more afraid of fungi now than there were in the past. So the question then becomes, why? There's this great book, you should buy it. It's about $150. But this is mine and Sarah's and Helen's kind of conceptualization of why the Anthropocene is driving things in favour of emerging fungal pathogens. Emerging fungal diseases really is a problem of our own making, and I'll be talking about two set pieces that illustrate this point. So fungi are wonderful eukaryotes. They've got highly flexible and adaptive elastic plastic genomes, so they really do respond to natural selection. And by golly, are we giving them selective arenas that they enjoy. So we've put 40% of the planet under the plough, and we coat this with monocultures of crops, and then the drugs that we use to control the organisms that like to use them, which are in the main fungi. We've bought, since I was in Berkeley 23 years ago, we've put 700 billion tons of carbon dioxide into the air, and this is causing noticeable greening. Warmer, wetter climates, that really biases things in a number of fungi's flavour. and then we just, we mix stuff up at quite an extraordinary level. I mean, a third of us will fly somewhere every year, and we're all going to be taking something with us, notwithstanding the kind of global trade that's going on. So we really mix things up. And this is where we start to see these driving signals, these signals of emerging fungi. So this was published in 2018, The Origins of a Chytrid, and it's an invasion story. This is an invasive disease, and the kind of backstory that I picked up from the conversations between David and Wake and John Taylor was that there was a plague of frogs. Well, actually, it was a plague on frogs, because the biblical plague was not being reciprocated here, but frogs were in deep trouble. And there were these key extinction events that were recognized in the 1980s, the gastric mouth-brooding frogs in Australia, and then this beautiful Costa Rican golden toad, which literally vanished in a single year. It was like an alien abduction that occurred. No corpses were ever found. And from these signature declines and losses in enigmatic environments, other declines and losses were seen across time. The mountain chicken frogs in Dominica and Montserrat, the Sierran frogs, Elytes in Europe, and then salamanders started going down the tubes. So what was going on? For the last 50 years, 500 species of amphibian had declined, and 90 are presumed extinct. The 90 presumed extinct is probably greater than that. But fully 40% of the world's amphibians are in decline. And this disease process is a major player. So it took a long time, but this zoosporic flagellate fungus was eventually identified and named by Joyce Longcore as Batrachochytrin dendromatosis, BD for short, causing amphibian chytridiomycosis. And it's a very simple swimming fungus. It infects the skin of frogs, and frogs have got very sensitive skins. They take in water, they take in air through their skin, and so you mess with their skin, then in some cases this can cause problems. Now, we also were blindsided by this second fungus. So, once is unlucky, but two starts to show some reproducibility. B cell came on the scene in the late 1990s when these fire salamanders in Europe, in the Netherlands, started getting rare very, very fast indeed. So this process was caused by a second chytrid, Chytrin salamandrivorans, and very rapidly, because there had been this kind of 20 years work on BD, we were able to go into natural collections of amphibians and frogs and to nail the reservoirs and where this had emerged from. And so B cell has its native range in Southeast Asia and had somehow, we don't know how, arrived in the Netherlands, spilled over to the fire salamanders there, and started driving these species extirpations. And B cell is quite happily chomping its way through the fire salamanders of Europe right now. So that mystery was solved in short order, but what actually was driving, what the backstory behind BD was far more enigmatic. In fact, I'd say it's one of the most elusive secrets in disease ecology. So we wanted to know where BD came from, and various kind of candidate source populations had been mooted: South Africa, North America, South America, Japan, Southeast Asia. There were all good reasons for believing any of these statements. And then when? So times of the most recent common ancestor for the chytrid ranged from 10,000 years ago to 100 years ago, and then how it spread across the planet. So at this time, Trent Garner, whose microsatellite protocol I used to such a good effect, had grown up into a raver in Zurich, where he was completing his PhD. And I hired him as my first postdoc, which was a very good idea because we've now published nearly 50 papers together. So it's been an amazing collaboration, but he was an absolute terrible postdoc to manage because he outgunned me in many respects, including age. So he very rapidly moved into a fellowship, and now he's a full professor University College London. But again, this is a great, it's a great piece of luck that my mind was prepared by my microsatellite experience to give Trent this job. And anyway, so he's a disease herpetologist, and we just knew that we had to get BD from across the planet, but we had to do this in a way that we didn't have to kill frogs. So we got very good at developing this toe clip protocol where you could go and snip off the end of an amphibian's digit, clean it, pop it into a tube of liquid media, get it back to the lab. And more than that, we developed this protocol in a way that we could farm it out to other people to get them to do the hard work for us. And that's the critical bit, because then people start sending you the isolates so that you can play with them. So we managed to do this across the planet and essentially hunt chytrids for genome sequencing. So again, this kind of distributed, outsourced method of doing biology is a real theme through my work, as you'll see. We also got money to go to fancy places. This is a trip to the Nourag base station in French Guiana, which is one of the largest contiguous unexplored rainforests in the world. We were sampling these poison arrow frogs here. Here's Priya working without gloves, not a good idea. We sat up all night cleaning our little toe clips and then popping them. And out of that, we maybe caught 800 amphibians over a 10-day sampling mission, of which we got 2 live chytrids out of it. So almost an expensive total failure, but not because we got those 2 isolates. So genome sequencing then went on, and we were able to define BD-GPL, a lineage that has emerged to colonize the Earth's amphibians. So here's the phylogeny here. Now, we didn't just recover one single clade, we discovered all this other wonderful diversity. So here was the African lineage, BD Cape, also found in the island of Majorca. BD Asia Brazil, this nice lineage that's found in the Brazilian Atlantic forests and across Korea, and then BD Asia 1, just found in Korea, and then this is GPL, and it's absolutely everywhere. So BD-GPL is what's been driving the majority of the Earth's declines. Not all of them, it turns out. BD Cape does have some action in Africa, but the extirpations are caused by GPL. And certainly when you throw it into amphibians and compare it against the lineages, in the main, it's highly virulent, although there is quite a lot of heterogeneity even in this phenotype that we're picking up now. This is where Trent comes in. He's very, very good at doing animal experiments. And when he started working for me, instead of isolating microsatellites, which is what I employed him for, he developed animal models, which enabled us to actually do a lot more, because genome sequencing came on very rapidly and made microsatellites redundant. So that's what was causing the global declines, BD-GPL. The global vector of BD turns out to be the amphibian trade. And we're very good at doing industrial farming of pretty much everything, and this turns out to include frogs. So this North American bullfrogs in Taiwan, and they're just a massive great soupy pool of BD-GPL, which then of course spills out into the local environments if you let it, and there's no biosecurity here. The global phylogeny was had great utility in actually picking out intercontinental links where BD had clearly been moved in very recent time, just because there were very little genetic diversity separating isolates. So we were able to map these extraordinary intercontinental translocations, and we were also able to pull out isolates of BD from amphibians that were in the trade. So we were able to show that in real time amphibians were being moved around with lineages of chytrid in them. So we found a lot of examples of this with GPL, this was being spread around by amphibians, but also all the other lineages. So this is BD Cape. You can see this wonderful translocation event here. This was a zoo that took African clawed toads into a captive breeding program in the Channel Islands, where it then spilled over into Aletes mulitensis, the Myopan midwife toad, which they were captive breeding to captively boost local populations on Majorca, where it was a very rare animal. So that crossover then seeded the island of Majorca with the African GPL. So, you know, completely unwitting bioinsecurity example. It was before BD was recognised, so it was a fairly ancient event, and this sort of stuff doesn't happen anymore. And then we also see the other lineages appearing in the trade. So when did BD-GPL emerge? Well, to actually get some more kind of focused time on that, we needed a lineage-specific, time-calibrated molecular clock with no recombination. And this is where the BD mitochondria came in great use, because it's huge. It's the biggest eukaryote mitochondria, 297 kilobases, and it's linear as well. It's not a circle. So it's a really unusual structure, but it's also it also has really nice clock-like behavior. So here we have isolates that we'd collected over time, and that's root to tip distance, and we were able to calibrate a nice mitochondrial clock off that, which dated the emergence of BD to the mid-20th century. So this really looked like an Anthropocene event. And look at the mitochondrial DNA phylogeny for all of those global GPLs there. I mean, they're just squished into this tiny little spot. I mean, so this really was the proof that we were looking for. And actually, when you burrow into certain areas of the nuclear genome, you're able to recover the same clock-like behavior. So it wasn't just a property of the mitochondrial DNA. So next week, Ben Shealy's big meta-analysis of global BD declines is coming out in Science, and it's a beautiful piece of ecological accountancy, I think we call it, because it sums the total of impact of chytrid on the planet's amphibians. So it's a really good evidence base for why we don't want to be letting this sort of process happen ever again. But Ben's declines click off in the late 1960s, early 1970s. So we've got these clocks are all coming together with the ecological data, which gives us confidence that we are actually correct. Southeast Asia appears to be the ancestral population of BD, and this is the set of evidence that we've mined out of the genomic data to pinpoint that blue lineage, the Asia 1 lineage, as containing the ancestral genetic diversity of all the global chytrids. They have a very flat Tajima's D compared to all the other lineages of chytrid. This is a marker of a disturbed population structure. When you see something that's a mutation, drift, equilibrium, then you're looking at some form of recombining ancestor. These Asia lineages all root basally to the rest of the lineages, which gives you some comfort that they are ancestral. They're also incredibly highly diverse. There's only 7 isolates of Asia 1, but that gives us the 39,000 SNPs compared to the GEL 423 reference compared to the other lineage. So it's a hyper-diverse population, and that presents very source-like behavior. And then finally, it's the alleles that you find in Asia 1 are donated to the other lineages of BD-GPL. So it really looks like Asia 1 and Asia is the ancestral diversity of BD-GPL. And that there have been multiple emergences which have caused that led to the global distribution of the other BD lineages. So this, of course, is Asia is the heartland for B cells, so we have another amphibian parasitizing chytrid there. They've been separated for 30 to 40 million years. But then you also see other fungi which have emerged out of Asia. So this was a Nature paper last year. The genome evolution across 1,000 genomes of brewers yeast. And their conclusion that genomic analyses support a single out-of-China origin for the species, followed by several independent domestication events. I mean, this is a very similar conclusion to what we've been making for BD. So I suppose the point being here is that Southeast Asia is a very diverse and interesting ecological biome with many fungi in it, some of which we need to be very cautious about. So we found that Southeast Asia is a likely heartland of chytrids, and it's the recent introduction into the amphibian trade through the auspices of the amphibian trade. We've also been kind of interested in the the heterogeneity that you see in the signal of these declines, because there's a common perception that if there's a chytrid recorded somewhere, then you're going to lose your frogs, and this is entirely incorrect. And so what this is doing is it's speaking to us about something that's underpinning the interaction between BD and the amphibians that it happens to be trying to invade. And so this kind of invasion process is really interesting with fungi because they're very good invaders on the whole. But the biotic resistance hypothesis postulates that successful invasion is negatively related to the diversity of the resident microbial community. So there's a lot of players there, you're going to find it hard to bust your way in. And we're, like others, are finding increasing evidence that this is true. And so a lot of my more focused lab work is field work is done in the Pyrenees in these absolutely wonderful pristine mountain lakes, which have enormous amounts of highly susceptible amphibians in there. Aletes obstetricans, the midwife toads. And so we've been going through time to these lakes and collecting animals and then looking at their infectious burdens. And what we've found is a series of lakes where the infection dynamics are very different. Some are epizootic and declining, and the frogs are becoming very rare, where others are surviving, in blue here. And these are the infectious burdens right here. So for 10 years now, I've been walking around these lakes, and here I'm just counting and the dead amphibians that I found. I have a peculiar speciality that I then put them into little lines and take photographs of them while my PhD students and postdocs do the hard work. But this kind of ends as these have become kind of very highly used images of the impact of a fungal invasion. So here's Kieran. He thinks he's a rock star. Here's a frog that is collected, which he'll then characterize the bacterial diversity on, throw it through a MySeq, and then see what's the diversity on these amphibians. And so what Kieran's been able to do is classify very different bacterial structures in these epizootic, declining versus enzootic surviving lakes. So in these this data fits the model that if you've got reduced bacterial diversity, you're more likely to have very high fungal dynamics occurring. So these enzootic lakes have high bacterial diversity. The epizootic lakes, it's very low, and there's some species of bacteria that are very characteristic, well-known pathogens that appear to be associated with these epizootic declining lakes. So it really looks like there's a specific kind of unique characteristic of these lakes which allows BD to flourish, invade, and to destroy an amphibian population. But if it's not right, then you don't get those epidemiological dynamics. So this is being used for this broader process of looking for bacterial probiotics that we might use to manipulate the environment in an amphibian's favour as opposed to the dreaded chytrids. And this is actually work that was kicked off in the Sierra Nevadas here by Cherry Van Spredenberg and Cherry Briggs's labs. And they recover very, very similar patterns to what we're finding in the Pyrenees. Low bacterial diversity associated with enzootic lakes, and where you have an epizootic, you've got high pathogen burden, then you've got a very different set of bacterial composition. And we're also extending this now to look at fungal species, and there seems to be associations there, the fungi that are associated with the chytrid. So the take-home point here is that the ecological outcome is very, very complex, and that these trans-kingdom interactions are going to be mediating the epidemiological dynamics that's superimposed on this broad global spread mechanism. How am I doing for time again?
42. That's 18.
Right. I'm just going to scamper. So I've talked about this origins of a chytrid. I'm now going to talk about artificial selection on the global scale. And this is a very different story, but here we have the high propagule pressure that's produced by a fungus, and we inhale these into our lungs. And when we inhale something into our lungs, it tends to be an aspergillus. There's probably 10 Canidia in every breath we take. Now, invasive aspergillosis infects huge numbers of people and is a very difficult infection to combat, unless you have an azole, in which case you could treat someone's infection. Now, the Anthropocene has given us some very useful drugs, but we deploy these on a gargantuan scale. And so this is a recent review that we did where we looked at what drugs we use and where fungi have evolved resistance to them. So these fungi do respond to the drugs we use, both for fungi that attack plants, but also for animals. Now what's important to this vignette is the azoles, which you can see are used both against plant pathogenic fungi and animals. So this is called dual use, and it's not generally a good idea. So again, another big meta-analysis done with Lady Sarah Gurr, showing that both human pathogens and plant pathogens are increasing in their resistance to azoles through time. So a very obvious temporal signal. Question being, how do these two intersect in humans? So this is if you want to read one thing that comes out of this lecture, it's Maren McKenna's article, Killer Tulips Hiding in Plain Sight. And this is referring to the fact that in certain parts of the world, we use incredibly high levels of fungicides. And certainly the bulb industry in the Netherlands is a prime culprit. So what we're looking for is how azole resistance occurs across Aspergillus fumigatus through time and space. So you can see we're kind of using that Chytrid methodology, but another system. So we've been sequencing global genomes from all these countries worldwide and using genomics to track azole resistance in time and space. So without going into the mechanism here, there's two azole resistance alleles, TR34 and TR46. And they occur very widely in the environment. So this is where you've got this Aspergillus fumigatus responding to selection by this agricultural azole. It also happens to be the same azole that clinicians use. So we've been sequencing way back into the 1920s and into the 40s, and you can see we've got no azole resistance here, now into the 1960s, 1980s, 2003, here we go, boom, the first incidence of TR34, 2010, we've got some more here, and then 2018. Now you can see that azole resistance appears to be global, but it's actually most of the actions here in Europe. And then there's a very obvious structure here, and only one incidence of azole resistance over there. So azole resistance is partitioned into this very strong population structure that we see in fumigators. So there's a whacking great genetic barrier here, which suggests that we're dealing with potentially two species of fumigators, but also why do you only see azole resistance occurring in this one clade? We really don't know the answer to this. So here we have clade B, clade boring, where we've only seen resistance once, and clade A, where it's very, very common indeed. So what we're trying to do here is collect the evidence base that connects these environmental occurrences of azole resistance to those that we find in patients. So we have those that we've recovered from patients. This is cystic fibrosis cohort in London, collected by Ali, in which 27% of azole-resistant infections are this environmental allele. And then we're recovering these from the environment of the United Kingdom and then sequencing their genome to see whether or not they're the same. At the same time, Jennifer Shelton has developed this really amazing way of sampling the entire country for what's in the air by sending out these little spore traps. It's a citizen science project, and her science solstices. So using a 1 8-hour time shot, appropriately organized through Twitter, she was able to get this distribution of the entire country and these blue spots of fumigatus. That's 1,400 colonies, 1,400. There's one PhD student, 1 8-hour time slot. And then, because Jen drinks, we don't actually know who Jen is. She's probably from another planet because she's so energetic. But she keeps on running these citizen sample scientist projects, autumn equinox, winter science solstice, and recovering. And this is Jen's big reveal. What's in the air in terms of the proportion of azole-resistant Aspergillus fumigatus? And it turns out to be between 3 and 4%. So I don't like this number. This is actually, I'm getting hit by an appreciable number of these propagules every day. So when we actually start sequencing these isolates, then we start finding that there are certain clones that are found in the clinic and the environment. You see, just from the UK, we have recovered the entire global diversity of this fungus. So this is a very, very mixed organism. But there is this one clade here that you see has got environmental and clinical isolates from all over the UK. So what we're sequencing from the lungs of a cystic fibrosis patient has come from the environment. This is direct evidence that we've got transmission for something that was pre-evolved to azoles. And this has real consequences for the patients because it means that their azole treatment isn't going to work. And by the time you've figured out that it isn't working, then you've lost the patient. So, where else are we finding this particular clone? We're finding it pretty much everywhere. It's in Europe, and then we're picking up it in Taiwan, in Thailand. And the distance between these European isolates that we find in leak fields and those in the Thai environment is only 500 SNPs. So this again is testament to how enormously we're mixing these populations of fungi. Now, what is kind of interesting to me is you've got azole resistance on a particular genetic background, clade A. What happens if that clade has got another trait that you don't want, like it's a faster grower, it does better, and because that means that you're then going to be hitchhiking another trait along with azole resistance, which is going to bias the global population of the most, one of the most numerous eukaryotes there are. So is there a difference between the two clades? And yes, there is. So this is clade A, growing in Aspergillus complete medium, and clade B. There's a lot of overlap, but these are actually significantly different traits. So here we have something that is being, the use of azoles will start biasing this population. This population is going to become more common compared to this one because of the background selection. And our clone that we're finding globally, which probably compromises 10% of Aspergillus diversity, is matching this high growth phenotype. So, we're exposed to these clones. There is an attack of the clones. They, when they do well, they do very, very well. BDGPL and then the Aspergillus superfit. So in conclusion, here we have the model of the Anthropocene. I do a little bit of stuff in Twitter, so I make sure that those tweets capture what I want to say. It's taken us a decade to pull this data set together and the message is clear. Moving wildlife around the planet is a very bad idea unless you know exactly what it is that you are moving, which we don't, so we shouldn't. I do not think we should be moving things around the planet like lizards and snakes into the pet trade. I just think that time has passed. And there'll be a lot of unhappy kids, but they can play with computers rather than animals. They do that already, don't they? I also think that the worldwide emergence of resistance to antifungal drugs does challenge human health and food security. And we've got a scales of balance here that we need to right, because we need food security, but we also need drugs that work in patients. So we do need better antifungal drugs, vaccines and diagnostics, and also stewardship, the use of what we've got as our populations grow. Because there's a lot of people coming through the pipeline. But that's what I believe. And in acknowledgement, I think this is I thought a little bit about this. And you can see this project. There are now hundreds of scientists involved in supporting these ideas. And they're not my original ideas, but I have a unique trait in that I can collaborate well. Why can I do that? Well, I've been supported through my life by all sorts of people. When I was a kid, a school teacher, friends and family. When I was a student, my fellow students and supervisors. And now that I'm a prof at Imperial, there's reviewers in the papers. Whoever thanks them for the work they do? It's a horrendous task, but we'll do it. The collaborators, the students that do all this great work, colleagues, mentors, staff, your head of department who protects you from the eye of Sauron. So, I think the point being here that it's an enormous support network that's required to support science. You also need the funders. And this is something that we tend to forget, because the system does actually make us be competitive but you have to play nice, don't you? Because if you don't, it's not going to work. And I think that this is the one thing that I learned in Taylor Lab, was that it takes a village to support this sort of work. And that I've been playing sort of nice ever since. Thank you very much for listening.
There is time for questions.
Can you speak a little to the relationship, the sort of chicken and egg question in the microbiome relationship to the BD infection? So, is it that the declining lakes have a different bacterial composition on the frogs because the BD has already gone through and wiped out a bunch of species, or is it that there are actually microbial composition is actually protected? Of course, we all want to believe that second point, but can you speak to your thoughts about which direction the causality?
There's a great observation conditioned on field trials, because we then have to reach into Trent Garner's animal labs to do the experiments. And so we make simple and we make complex environments, and we put the animals in.
You paint an animal with.
No, you can rear them different substrates that assemble a different community. So we're going to work with Tom Bell and actually use robots to build these, and we've done a little bit of work on that. But it appears to be that, I mean, the environment, if you do that sort of experiment, then the infection dynamics are completely different. So the prior occupancy of those microbial species does determine the outcome. I'm not sure that's what's happening here, though. I mean, I think probably both situations are occurring. But certainly some of these lakes, they were just ecologically different anyway, and that they did have different properties that selected different bacterial species, but they were all there. So there's other layers of this. I think it's ecologically incredibly complex.
Cool. Dave?
We know that BD moves around on its hosts. So if you move the host around, you move BD around. But how does BD move around by itself? And how does it get to really remote places? Where there been no introductions of
like French Guiana.
Yeah. How does it get there?
Well, there's that, that 100-kilometer-a-year figure from the episodic wave down through Central America. Yeah. It's very, very fast for a frog, that isn't it? It's too fast. So Anne and Frank in Belgium have done these experiments showing that you have zoosporangia growing on toenails of water bird feet. So they are very keen on the water birds can at least do some limited dispersal. They're probably very rare. I mean, BD is incredibly infectious when it gets somewhere. So perhaps that entirely rare, highly stochastic event is what's necessary. So I think there are other vectors. Jonathan Colby, pointed towards, the aerial column, some wind dispersal. I don't know, but I think that happens.
They dry out really easily too, and they're not very tough.
Well, B-cell is very tough because it has this extra zoospore that they discovered. But BT is an incredibly fragile organism. It's incredible to me that it is as successful as it is. So, and certainly at Asilomar, they're now showing that zoospores actually crawl, so they produce pseudopodia, and they think that this is a mechanism to actually allow almost instant reinfection of a host. So it might be that it actually has two dual traits that are timed for different times in its zoosporic life cycle. A local side for reinfection of your host, and then perhaps some more resilience which allows longer distance dispersal. There's a lot of BD biology that we're only getting to grips with now through cell biologists.
So I have a question. So the global pathogenic lineage at one point was thought to be a hybrid. And is that still the thinking? And then what's the ploidy of the ones in Asia A?
So it might be an old, well, it might be an old hybrid, so it's a little hard to tell. We find new hybrids dotted around where these lineages have come together. So there's some really nice ones in Brazil, but there's also nice ones in Southern Africa. But actually GPL just seems to be a random pick of that Asia 1 diversity. So most likely not a hybrid, and they're all got this strange multi-ploidy. So no haploids there.
So they're not just one ploidy either.
No.
Okay. It's like Allomesis. If there are no other questions, I have. Oh, go ahead.
For about the patchy infection of the frogs around the world, the ones that are asymptomatic but are infected.
Like North American bullfrogs.
Sure. Yeah. Are they? What's the load, like the load of the bacteria on the individual? Are they covered in these fungi, or the frogs like mounting a resistance, and there's like a small population on the frog?
Yeah. So what determines whether an amphibian survives or tolerates, is a better word, a BD infection is what antimicrobial peptides it produces, and so good antimicrobial peptides will control the BD infections. So that's one component, but then, and that's also got a dietary component as well, because you actually have to feed on things like ants to produce these AMPs. Tetrodotoxin is on amphibian skins and will have some anti-BD activity. So again, it's incredibly complex. But there will also be those resident microbacteria, and a particular frog will have selected for its own particular set of commensals, and that will also, I haven't talked about the mycobiome. We have a big project where we're mapping the fungi that are associated with amphibians around the world. And some, are negatively associated with BD. If you have this fungus, then you don't have BD. So all of these trans-kingdom interactions are playing out on the background of these these very complex molecules that are produced by fungi, by the host, and by the players themselves. So it's complicated.
Let me make one comment before the last question. You are all welcome to follow us to Koshland Hall, to room 338 on the third floor for a reception for man fishing.
With respect to your tweet about not spreading things around before we know what they're doing, can you comment on prospects for environmental probiotics, like what you mentioned with your protective lake being a probiotic, like the microbial community being a protective lake there? And what if it is actually a pathogen that's another organism
So there have been, the bacteria that produces violacin, you're not allowed to move a bacterium to another part of the world. So if you're interested in a compound that has got anti-BD properties like violacin, you have to re-isolate that bacterium from the source. So that was done in Panama by Brian Gratwick's group. So they took Van Spredenburg's original observation, re-isolated the bacterium in Panama, then applied it to Atelopus zatiki, but they all died. So this, even when you've dipped your frog in yogurt and then released it, it doesn't. There's no persistence really. To actually kind of drive your particular probiotic into a complex thing like an amphibian skin hasn't yielded a good result yet. So there may not be legs in that approach. But it would be really nice to be able to change the environment. And there are also the micropredator stories. So if you boost the number of cladocerans in the water, then you'll comb out the zoospores. So that's that. It would be nice to kind of improve the ecosystem for them. Then you might be able to control BD. And certainly in the bromeliads, you don't get high rates of BD infection because they're full of rotifers that are chomping BD as fast as they're produced by frogs. So there seems to be some natural kind of processes going on that mimic that. But as to be able to do that as humans, no. So just don't spread the stuff in the first place.
Are there viruses that colonize BD or Aspergillus that have been characterized or not?
Yeah, so there are microviruses in Aspergillus, and that would, at the cinema there was a lot of conversation about actually looking for some of those, because they especially the RNA viruses do have biocontrol potential. For Chytras we've had one look and we didn't find anything, but there's new bioinformatic processes. You can take the deep sequencing data and start to explore for the DNA viruses now without just having to run out and look for that big bright band on the gel. So I think all of this needs to be redone in the kind of short read data sets that we've got. what's that kind of DNA or RNA dark matter that we haven't assembled. We'll find it there.
So you're all invited to continue the discussion in the third floor of Schossner Hall. Thank you.